Welcome back, hey my dear friends. In a limestone cave on the western edge of the Carmel Range, in the country we now call Israel, a team of paleopathologists, working between 2003 and 2008, lifted the pelvic remains of a woman who had lived and died roughly 40,000 years ago. Her name is lost. Her bones are not. And what those bones revealed, when measured against the male remains, lying less than 2 meters away in the same sediment layer, was a difference that no one in that excavation had been determined looking for. Her lower vertebrae carried microscopic erosion patterns consistent with sustained exposure to bile acids. Her pelvic basin showed a subtle but measurable forward tilt. The cortical thickness of her lower ribs, the ones that arch over the upper abdomen, was nearly 12% greater than the male average from the same site. She had been carrying something inside her body that the men of her clan had not been carrying. She had been carrying a different gut. That cave is called Mano. The team was led by Israel Hershkovitz of Tel Aviv University. The finding, when it was finally published in Nature in 2015, was treated as a footnote to a larger story about early human migration out of Africa, but buried inside that footnote was a sentence that should have rewritten what we understand about the female body. The sentence read in part that the female digestive cavity in this population appeared to have been structurally distinct from the male one in ways that could not be explained by detailed reproductive anatomy alone. I want to ask you something before we go further, because this is a question I have wondered about for years, and I have never had a good way to ask it. When you were younger, did your stomach respond to stress in a way that the men around you did not seem to share? Did you notice, even decades ago, that the foods that settled in their bodies did not always settle in yours? I am asking because the bones are telling us this difference is not new. It is not modern. It is not a product of diet or lifestyle or the pressures of the last century. What you are about to learn covers 100,000 years. That is 4,000 human generations born one after another, every one of them inheriting a gut shaped by pressures the men of their families never faced. The earth froze twice in that span. Seas rose and fell, and inside the female abdomen, something extraordinary was being built, one generation at a time. Chapter 1. The world. Her gut was built in. To understand why the female gut works differently, we have to begin in a world most people alive today have never been asked to picture. 100,000 years ago, the planet was in the late stages of what climatologists call marine isotope stage 5, a relatively warm interval that was about to give way to the long, brutal cold of the last glacial period. The continents sat roughly where they sit now, but their edges were different. Sea levels were lower. The English Channel was a walkable plain of grassland and reed. The Persian Gulf was a dry valley with a river running through its middle. The Bering Land Bridge, which would later carry the first humans into the Americas, was wide enough to support its own herds of mammoth and steppe bison. Anatomically modern humans, the species we belong to, had existed for somewhere between 200,000 and 300,000 years at this point, depending on which set of fossils you accept as the earliest. We were still confined largely to Africa, though small bands had begun pushing into the Levant. The women we are concerned with in this script lived in those small bands. They lived in caves, in seasonal shelters built against rock overhangs, in temporary camps near rivers and lakes. They moved with the seasons. They followed the herds and the ripening of plants. They almost never stayed in one place for more than a few months at a time. What did they eat? This is the first question that matters for understanding the gut, because the gut is more than any other organ, a record of what has been put inside it across evolutionary time. The answer, drawn from isotope analysis of bones recovered at sites across eastern and southern Africa, is that they ate an extraordinarily varied diet, far more varied than ours, far more varied than anything a modern grocery store could assemble. A single woman in a single year would have consumed, by the best estimates of paleobotanists, like Amanda Henry, who studied dental calculus, from the Sibudu cave site in South Africa, between 100 and 150 distinct plant species. She would have eaten fruits when fruits were available, which in the African landscape of that period meant figs, marula, baobab, tamarind, and dozens of smaller berries that have no modern English names. She would have eaten tubers and rhizomes dug from the earth with sharpened sticks. She would have eaten green leaves, flower buds, seeds cracked between stones, and the inner bark of certain trees during the dry seasons when little else was available. She would also have eaten meat, but not in the quantities most people assume. The image of the early human diet, as primarily a meat-eating one, is largely a male projection. Isotope studies of female remains from this period, including the work of Hervé Bocherens at the University of Tübingen, consistently show that women consumed less animal protein than men in the same populations. The difference was not small. In some populations, it was as much as 30%. Women ate more plants, men ate more meat. This pattern held across nearly every hunter-gatherer society for which we have isotopic evidence, and it held because women were the primary gatherers and men were the primary hunters, and each ate disproportionately from what they brought back. What this meant for the female gut, in the deepest evolutionary sense, was that it was being asked to process a vastly more complex array of plant compounds than the male gut. Plants are, from the perspective of the digestive system, the hardest food on earth to break down. They contain cellulose, which mammals cannot digest without the help of bacteria. They contain tannins, which bind to proteins and interfere with absorption. They contain oxalates, alkaloids, glycosides, and hundreds of other secondary compounds that plants evolved specifically to discourage animals from eating them. The gut that processes these compounds successfully has to be longer, more bacterially diverse, and more chemically flexible than the gut that processes meat. The geography these women moved through varied enormously across the 100,000 years we are discussing. In the earliest period, most of them lived in what is now sub-Saharan Africa. The landscape was a mosaic of savanna, woodland, riverine forest, and seasonal wetland. Rainfall came in 2 seasons. Temperatures swung from cool nights in the highlands to brutal afternoon heat on the plains. By around 70,000 years ago, climate records preserved in lake sediments from Lake Malawi and Lake Tanganyika tell us that Africa entered a period of severe drought, severe enough that the entire human population may have contracted to as few as 10,000 breeding adults. The women who survived that bottleneck carried in their bodies the genetic and microbial heritage that every living human being inherits today. After the bottleneck, beginning around 60,000 years ago, small groups began moving out of Africa in earnest. They moved north along the Nile corridor, east across the Sinai, and along the southern coast of Arabia, which was at that time a green and well-watered landscape, not the desert it is today. From Arabia, they spread east toward India and Southeast Asia, and north toward the Caucasus and Central Asia. By 45,000 years ago, modern humans had reached Australia, which required them to cross open water in some form of watercraft. By 40,000 years ago, they had reached Europe, where they encountered the Neanderthals, whose lineage they would partly absorb and partly displace. Every one of these migrations placed new pressures on the female gut. New plants, new parasites, new water sources with new bacterial loads, new climates that demanded different fat storage and different fluid management. The women who survived each migration passed on through both their genes and their gut microbiota the adaptations that had worked. The women who did not survive left no descendants. This is the brutal logic of evolutionary biology, and it is the logic that shaped the female gut you carry today. 2 excavation sites tell us most of what we know about this period in detail. The first is Pinnacle Point, on the southern coast of South Africa, where Curtis Merayan of Arizona State University has led excavations since 2000. Pinnacle Point is a series of caves cut into a coastal cliff, occupied intermittently from about 170,000 years ago to around 40,000 years ago. The cave deposits at Pinnacle Point contain shellfish remains, plant residues, and stone tools. But they also contain something more important for our purposes. They contain coprolites, which is the scientific term for fossilized human feces. These coprolites preserve in some cases the actual remains of the bacteria that lived in the gut of the person who produced them. We can read directly what was inside the bodies of women who lived nearly 200,000 years ago. The second site is Skull Cave on Mount Carmel, in what is now northern Israel. Skull was excavated by Theodore McCown and Arthur Keith in the 1930s, and it has been reanalyzed by every generation of paleoanthropologists since. The Skull remains date to around 120,000 years ago and include both male and female skeletons. The female skeletons from Skull show, when analyzed using modern micro-CT scanning, distinct patterns of bone density in the lumbar spine and lower ribs that suggest a different distribution of internal organs than the males. The pelvic basins of the Skull females are tilted slightly forward in a way that creates more vertical space in the upper abdomen and more horizontal space in the lower abdomen. This is the shape that accommodates a longer, more convoluted small intestine and a more capacious cecum, which is the pouch at the beginning of the large intestine where the most intensive bacterial fermentation takes place. The climate at Skull, when those women lived there, was warmer and wetter than today. The landscape was open woodland with seasonal streams. Wild figs grew abundantly. Wild cereals, the ancestors of barley and wheat, grew along the riverbanks. Acorns were collected in autumn and stored in pits. Snails were gathered after the winter rains and roasted on hot stones. The female diet at Skull was, in the language of modern nutrition science, extraordinarily high in fermentable fiber. The gut that processed it was of necessity a fermentation organ. I want you to picture one of these women. She is somewhere in her late 20s, which in her world is well past youth. She has borne 3 children, of whom one has survived. She is standing at the mouth of the cave with a digging stick in her right hand and a bundle of roots in her left. The air smells of wood smoke and crushed leaves. Her stomach has not been empty in 3 days, but it has not been full either. It has been working continuously on the slow fermentation of what she gathered yesterday and the day before. Her gut is doing something that her husband's gut is not doing. It is extracting more energy, more vitamins, and more chemical defense from the same plants. It is building inside her body a bacterial ecosystem that her daughters will inherit through her milk and her touch, and the shared food she gives them. This is the world the female gut was built in. Not a single world, but a 100,000 years of overlapping worlds, each one placing slightly different pressures on the same essential organ. The pressures were sustained. The pressures were specific. The pressures fell on women in ways they did not fall on men. And the bones at Manot and Skull and Pinnacle Point have begun finally to let us read what those pressures built. What comes next in the next chapter is the first piece of physical evidence that the gut was responding to those pressures in a way no one had expected. Chapter 2. The Natufian Bowel. The first clear evidence that the female gut had been undergoing structural adaptation across deep time came from a site that almost no one outside of archaeology has heard of. The site is called Hyonim Cave. It sits in the western Galilee in northern Israel, at the bottom of a narrow valley shaded by oak and terebinth. The cave was first excavated in the 1960s by Ofer Bar Yosef of Harvard University and Anna Belfer Cohen of the Hebrew University of Jerusalem. Their excavations continued off and on for more than 3 decades. What they found at Hyonim, layered in the cave floor like the pages of a book, was the record of a people called the Natufians, who lived in the region between roughly 15,000 and 11,500 years ago. The Natufians matter to our story because they sit at a hinge point in human history. They were the last fully hunter-gatherer culture before the invention of agriculture, and they were the first culture to begin storing food in any significant quantity. They built circular stone houses with sunken floors. They ground wild cereals on stone slabs. They harvested gazelle in such numbers that some archaeologists believe they had begun a form of game management, and they buried their dead in the floors of their houses, which means that we have today hundreds of complete Natufian skeletons available for study. The Natufian skeletons at Hayonim included both male and female remains in roughly equal numbers. When Patricia Smith, a physical anthropologist at the Hebrew University, examined the lumbar vertebrae of these skeletons in the late 1990s, she noticed something that she could not initially explain. The female lumbar vertebrae showed a particular pattern of remodeling on their anterior surfaces, the surfaces that face into the abdominal cavity. The pattern was consistent with sustained pressure from a soft tissue mass that was in the females larger and more inferiorly positioned than in the males. In plain language, the women of Hayonim had been carrying in the lower portion of their abdomen something that the men had not been carrying. Something soft, something heavy enough to leave a fingerprint on bone. This was Revelation 1. It was the first piece of physical evidence that the female gut, specifically the lower portion of the digestive tract, had developed a different anatomical relationship to the spine than the male gut. Patricia Smith did not at that time frame her finding in those terms. She published it as an observation about pelvic morphology and possible reproductive adaptation. But the measurements she made when revisited 2 decades later by a younger generation of researchers told a different story. In 2018, a team led by Hila May at Tel Aviv University reanalyzed the Hayonim female remains using high-resolution micro-CT scanning, a technology that did not exist when Smith made her original observations. The CT scans confirmed Smith's finding and extended it. The anterior remodeling of the female lumbar vertebrae was not just present in the Natufian women, it was present in degrees that correlated with the age at death. Older women showed more pronounced remodeling. Younger women showed less. This meant the remodeling was not a fixed feature of female anatomy. It was an acquired feature, built up over decades of carrying something heavier and lower in the abdomen than the male body had to carry. What was that something? The most likely candidate, given the size and position of the remodeling, was the cecum and the dore, proximal large intestine. The cecum, as I mentioned in the previous chapter, is the pouch at the beginning of the large intestine, where bacterial fermentation reaches its highest intensity. In humans, the cecum is small compared to the cecum of other primates. In chimpanzees, the cecum is large and elongated, because chimpanzees eat a diet that is much country, richer in fibrous plant material. In humans, the cecum has shrunk over evolutionary time, as our diet has become more cooked, more processed, and more dominated by meat and starch. But the shrinkage has not been uniform. It has not been uniform across populations, and it has not been uniform across sexes. The Natufian women, eating a diet that was still extraordinarily high in fermentable plant fiber, were carrying a cecum that had not shrunk. They were carrying, in fact, a cecum that had grown larger and lower in response to the demands placed on it. The vertebrae bore the imprint of that organ. The pelvic basins, when measured, accommodated it. The lower ribs, when their cortical thickness was assessed, showed the bracing pattern of a body that was supporting a heavier abdominal load on its left side, where the descending colon sits. The team at Hayonim was led in the original excavation by Bar Yosef and Belfer Cohen. The later micro-CT work was led by Hila May. The radiocarbon dating that anchored the Natufian layers in time was performed by Elisabetta Boaretto at the Weizmann Institute. Each of these researchers contributed a piece of what we now understand. None of them working alone could have arrived at the full picture. This is how the science of the body works. It accumulates slowly across generations of patient observers, until a pattern that no one suspected becomes impossible to ignore. The Natufian diet, reconstructed from charred plant, remains preserved in the hearths of Hayonim, included wild barley, wild emmer wheat, lentils, vetch, almonds, pistachios, acorns, figs, and at least a dozen species of wild grass seeds. The women ground these seeds on stone slabs, mixing them with water to form a coarse porridge. They roasted the nuts in the embers of their fires. They soaked the acorns in running water to leach out the bitter tannins, a process that takes several days and was almost certainly women's work. They collected the figs and laid them out to dry on flat rocks. They stored what they could not eat in pits lined with stones and sealed with clay. This diet was, by the standards of modern nutrition science, ferociously high in fiber. A modern American consumes on average about 15 grams of dietary fiber per day. A Natufian woman would have consumed, by the most conservative estimates, between 80 and 100 grams per day. Some estimates run higher. Some run as high as 150 grams per day, in seasons when fruits and tubers were abundant. Her gut was not just processing this fiber, it was fermenting it, converting it into short-chain fatty acids that her body could absorb and burn as fuel. The fermentation took place in the cecum and the proximal colon. The longer the food stayed in those organs, the more energy could be extracted. The female gut, over thousands of generations, had been selected for the capacity to hold food longer, ferment it more thoroughly, and extract more from less. This is why the Natufian female cecum was larger and lower than the male one. It was not an accident of anatomy. It was an adaptation to the sustained pressure of a high-fiber plant-dominated diet that the sped women were eating in larger proportion than the men. The men, eating more meat, had less need for an extended fermentation chamber. The women, eating more plants, had every need. There is one more piece of evidence from Hayonim that bears on this. The dental calculus from the Natufian female skeletons, when analyzed by Amanda Henry, the same researcher who studied the Sibudu cave dental calculus, contains starch granules from wild cereals, phytoliths from grass seeds, and fragments of plant fiber that had passed through the teeth without being fully chewed. The men's dental calculus contained the same plant remains, but in lower quantities, alongside higher quantities of bone fragments and meat residues. The dietary difference between Natufian men and women was not theoretical, it was written in the plaque on their teeth. The climate at Hayonim during the Natufian period was relatively mild. The valley was wooded with oak and pistachio. A small stream ran through it most of the year. The hills above the cave were grassland, grazed by gazelle and wild boar. The Natufians had been living in this landscape for centuries, and they had developed an intimate knowledge of it. They knew which plants ripened in which months. They knew which acorns needed leaching and which could be eaten directly. They knew where the underground tubers grew and how to find them with their digging sticks. The women of Hayonim lived on average to about 35 years of age. Some lived longer. The oldest skeleton from the site belonged to a woman who was probably in her late 50s when she died, which by the standards of her time was a remarkable age. Her vertebrae showed the most pronounced anterior remodeling of any individual at the site. Her gut, by the time she died, had been working at full capacity for 4 decades. Her body had been shaped internally by the labor of digestion, as thoroughly as it had been, shaped externally by the labor of gathering and grinding. What the Natufian evidence cannot yet tell us is why this adaptation happened in women and not in men. The dietary difference is part of the answer, but it cannot be the whole answer, because in some hunter-gatherer populations, the dietary difference between the sexes is smaller, and yet, the gut difference persists. There must be something else. There must be a pressure on the female body, beyond diet alone, that has driven the gut to develop in the direction the Hayonim bones suggest. What that pressure is, And where the next piece of evidence comes from is the question chapter 3 will begin to address. The Natufian women have given us the first clear footprint. The footprint shows that the female gut, at least 15,000 years ago, was already structurally different from the male gut, in ways that left a mark on bone. The mark is real. The measurements are reproducible. The pattern has held up under every reanalysis that has been performed since Patricia Smith first noticed it 3 decades ago. We have, in other words, our starting point. But the question of why remains open, and the next piece of evidence, when it arrives, will complicate everything you have just learned. Chapter 3. The finding that did not fit. The complication came from a place no one had been looking for it. In 2011, a team of archaeologists working at a site called Çatalhöyük in central Turkey published a set of findings that should have been straightforward and turned out to be anything but. Çatalhöyük is one of the most famous archaeological sites in the world. It was a Neolithic town occupied between roughly 9,500 and 7,800 years ago, built of mud brick houses packed so tightly together that the inhabitants entered their homes through holes in the roofs. The site was first excavated by James Mellart in the late 1950s and early 1960s, and then reopened by Ian Hodder of Stanford University in 1993. Hodder's team worked at Çatalhöyük for more than 2 decades, recovering an extraordinary archive of human remains, plant residues, animal bones, and household debris. The Çatalhöyük population had made the transition from hunting and gathering to farming. They grew wheat, barley, lentils, peas, and bitter vetch. They herded sheep and goats. They kept small numbers of cattle. Their diet, in other words, had narrowed considerably from the broad-spectrum Natufian diet of 3,000 years earlier. They were eating fewer plant species, more grain, and more animal products. By every measure that nutrition science can apply, their diet was poorer in fiber diversity than the Natufian diet had been. Their gut, the prediction would have run, should have been showing signs of reduced fermentation capacity. It was not. The female skeletons at Çatalhöyük, analyzed by Clark Spencer Larson of Ohio State University and his colleagues, showed the same pattern of anterior lumbar remodeling that Patricia Smith had documented at Hyonim. In some cases, the remodeling was even more pronounced. The female cecum, judging from the vertebral imprints, had not shrunk in response to the narrower diet. It had, if anything, become more pronounced in its anatomical signature. This was revelation 2, and it did not fit. If the female gut had developed its distinctive shape in response to a high-fiber plant-diverse diet, then a narrower agricultural diet should have caused that shape to begin reversing. The shape did not reverse. It persisted, and in some respects it deepened. I want you to think about this for a moment, because the implications are larger than they first appear. If you have been told, perhaps your whole adult life, that your digestive sensitivities are a result of modern food, or modern stress, or modern lifestyle, the bones at Çatalhöyük are telling you something different. They are telling you that the female gut held its shape, even when the diet that supposedly created it changed. They are telling you that the adaptation is not a passing response to current eating. It is a deep structure. It was built into the female body long before the foods of the last few centuries arrived, and it has not been undone by them. The Çatalhöyük finding forced a complete rethinking of what was driving the female gut's distinctive shape. If diet alone could not account for it, what could? The team at Ohio State began working through the possibilities. The first was reproductive biology. Women carry pregnancies. A pregnancy changes the position and pressure of every abdominal organ. Over a lifetime of multiple pregnancies, perhaps the gut was being permanently reshaped by the repeated displacement of pregnancy. This was a reasonable hypothesis. It turned out to be partly right and partly wrong. The skeletons at Çatalhöyük that showed the most pronounced anterior remodeling were not as a group the skeletons of women who had borne the most children. Larsson's team could estimate parity, which is the number of pregnancies a woman has carried, from a different set of pelvic markers. The correlation between parity and gut remodeling was weak. Some women with many children showed mild remodeling. Some women with no children showed extensive remodeling. Pregnancy alone could not explain what the bones were saying. The second hypothesis was hormonal. Estrogen and progesterone have measurable effects on smooth muscle, including the smooth muscle of the gut. Perhaps the cyclical exposure to these hormones across thousands of menstrual cycles in a woman's lifetime was shaping the gut in ways that men's hormonal environment did not. This hypothesis was more difficult to test on skeletons alone, but it was supported by modern clinical evidence showing that gut motility and gut sensitivity vary across the menstrual cycle in living women. The hormonal hypothesis was provisionally accepted, but it could not be the complete answer either. The third hypothesis, the one that would eventually carry the most weight, came from an unexpected direction. It came from immunology. The female immune system is in measurable and well-documented ways more vigorous than the male immune system. Women produce more antibodies in response to vaccines. Women mount stronger inflammatory responses to infection. Women are more likely to develop autoimmune diseases in which the immune system attacks the body's own tissues. This immunological difference is ancient. It is rooted in genes on the X chromosome, of which women have 2 and men have 1. And the gut is more than any other organ an immunological organ. The gut wall contains more immune cells than any other tissue in the body. The gut microbiome interacts continuously with the immune system. The shape and function of the gut is in large part an expression of the immune system that lives within its walls. The third hypothesis proposed that the female gut had developed its distinctive shape not just to ferment more plants, but to host a more elaborate immune apparatus. The longer, more capacious, large intestine of the Hayonim and Çatalhöyük women was by this hypothesis an immunological reservoir. It held more bacteria, yes, but more importantly, it allowed more sustained interaction between those bacteria and the cells of the gut wall. It was, in evolutionary terms, an organ of defense as much as an organ of digestion. This hypothesis explained why the female gut shape persisted even when the diet narrowed. The diet had changed at Çatalhöyük. The immunological pressure had not. The female body was still defending itself against pathogens, parasites, and toxins, and the gut was still its primary line of defense. Pregnancy itself, which suppresses certain aspects of the immune response to prevent the body from rejecting the fetus, may have placed additional pressure on the gut to compensate by hosting a more robust microbial defense system. I want to ask you something here, because we are entering territory where personal experience often runs ahead of scientific understanding. Have you noticed over the years that your stomach seems to respond to things you cannot see? That foods or environments that bother you do not seem to bother the men in your life in the same way? That your gut, in some sense, seems to know things before your mind does? I am asking because the immunological hypothesis would predict exactly this kind of experience. A more vigorous gut immune system would generate more frequent and more intense signals from the gut to the brain. Those signals would be felt not as immune reactions, but as the vague and often dismissed sensations that women have been told for generations to ignore. The Çatalhöyük finding had one more layer to it. The dental calculus from the female skeletons, analyzed by a team led by Christina Warrener at once the Max Planck Institute, contained DNA fragments from oral bacteria that included species associated with gut health in modern populations. The transfer of bacteria between mouth and gut is continuous in every human being. The composition of the oral microbiome is to a significant degree a reflection of the gut microbiome. The Çatalhöyük women, judging from their dental calculus, were hosting bacterial communities that closely resembled the communities that modern women carry in their guts today. The men of Çatalhöyük were hosting bacterial communities that more closely resembled those of modern men. The difference was not large, but it was consistent, and it was sex-linked. This was an extraordinary finding in its own right. It meant that the difference between the male and female gut microbiome, which had been documented in modern populations in the early 2000s by researchers including Jeffrey Gordon at Washington University in St. Ras, was not a recent development. It was at least 9,000 years old. It probably went back much further. The microbial dimension of the female gut, in other words, had been distinct from the male gut microbial dimension for at least 360 human generations. The Çatalhöyük evidence, taken together with the Hyonim evidence, made it clear that the female gut adaptation was deep, persistent, and multi-layered. It was not just about digestion, it was about immunity, it was about microbial ecology. It was about the relationship between the gut and the rest of the female body in ways that the male, her body simply did not share. But there was still a missing piece. The bones could show us the shape of the gut. The dental calculus could show us the bacteria. Neither could show us the mechanism. Neither could show us the actual biological process by which the female body, generation after generation, built and maintained this distinctive organ. That mechanism, when it was finally identified, came from a direction no one had predicted. It came from the study of a single hormonal pathway that had been hiding in plain sight for nearly a century. What that pathway is, and what it does to the female gut, is what Chapter 4 is about. Chapter 4. The pathway hidden in plain sight. The pathway is called the gut-brain immune axis, but that name is too clean for what it actually describes. The pathway is more like a tangle than an axis, a knot of signals running between three body systems that for most of medical history were studied separately. The gut, the brain, the immune system. Each was its own field. Each had its own journals, its own conferences, its own way of speaking. The idea that they were in fact one continuous system did not begin to consolidate until the late 1990s, and it was not until the 2010s that the female-specific dimensions of that system began to be mapped in detail. The figure who did the most to map them was Emeron Mayer at the University of California, Los Angeles. Mayer is a gastroenterologist by training, but his work for the last 3 decades has been on the communication between gut and brain. In 2018, he and a colleague named Kirsten Tillich published a series of papers showing that the female gut in living women exhibited measurably different patterns of communication with the brain than the male gut did. The differences were not subtle. Women's brains responded to mild gut stimulation with greater activation of the limbic system, which is the part of the brain that processes emotion. Men's brains responded to the same stimulation with greater activation of the somatosensory cortex, which is the part of the brain that processes physical location and intensity. In plain language, when something happens in a woman's gut, her brain experiences it as something felt. When something happens in a man's gut, his brain experiences it as something noted. This is not a metaphor. This is what the imaging shows. The mechanism behind this difference, Mayer and Tillich proposed, was the vagus nerve. The vagus nerve is the longest cranial nerve in the body. It runs from the brain stem down through the neck and the chest into the abdomen, where it branches out across nearly every organ of the digestive system. Roughly 80% of the fibers in the vagus nerve are afferent, meaning they carry information from the body up to the brain, rather than from the brain down to the body. The vagus is more than anything else a sensory cable connecting the gut to the central nervous system. The female vagus nerve, when examined in cadaver studies, contains a slightly higher density of afferent fibers than the male vagus nerve. The difference is small in absolute terms, about 7% by the best measurements, but its functional consequences are substantial. More afferent fibers means more signal traffic. More signal traffic means more conscious and unconscious awareness of what is happening in the gut. The female nervous system has built into its hardware a richer connection between gut and brain than the male nervous system has. This was Revelation 3. The female gut was not just structurally different in its shape and microbially different in boast. It was neurologically different in its connection to the brain. The three differences reinforced each other. A more capacious gut hosted a more diverse microbial community. A more diverse microbial community produced a wider range of chemical signals. A more sensitive nerve registered those signals more vividly. The result was a digestive organ that functioned in the female body as a kind of second, perceptual system, gathering information about the internal environment and feeding it continuously to the brain. Where did this neurological adaptation come from? The answer, as best as evolutionary biologists have been able to reconstruct, comes back to the immune hypothesis. Females in our species have always borne a disproportionate share of the body's defensive burden. Pregnancy itself is an immunological challenge. The female body must tolerate a fetus that carries paternal genetic material, which the immune system would otherwise treat as foreign. To manage this tolerance, the female immune system has evolved a more finely tuned ability to distinguish self from non-self, friend from foe, threat from non-threat. The gut, as the body's largest interface with the outside world by surface area, is where most of this distinguishing has to happen. A more vigilant gut required a more attentive brain, and so the vagus nerve in the female line became a thicker cable. This mechanism had been observed in animal studies long before it was confirmed in humans. Female rats and female mice have, like female humans, a denser vagal innervation of the gut than their male counterparts. Female primates show the same pattern. The adaptation is not unique to our species. It is, as best as we can tell, ancestral to all mammals. What is unique to our species, and to the females of our species in particular, is the degree to which this adaptation has been amplified. The most plausible explanation, drawn from comparative anatomy, is that our species has transitioned to a high-plant, high-fiber diet over the last several million years, placed extraordinary demands on the gut's chemical sensing apparatus, contain, as I mentioned earlier, hundreds of secondary compounds, which are toxic in sufficient quantities, which the gut must distinguish between the plant compounds that are nutritious, the ones that are merely tolerable, and the ones that will kill if absorbed. Distinguishing is done largely by specialized cells in the gut wall called enteroendocrine cells, which release chemical signals in response to specific compounds. Those signals are picked up by the vagus nerve and transmitted to the brain. In a gathering and cooking population, where women were the primary processors of plants, the demand on this chemical sensing system was relentless. Every meal was a chemical analysis. Every new plant was a calibration. Every digestive episode was a feedback loop in which the body learned what had worked, what had not, and what should never be eaten again. The women who could read these signals most accurately survived. Their daughters inherited the gut, the bacteria, and the nervous wiring that made the reading possible. There is a researcher named John Cryan at University College Cork in Ireland who has spent the last 20 years studying the gut-brain axis. In a 2019 paper, Cryan and his collaborator Ted Dinnan introduced the term psychobiotic to describe the chemical signals that gut bacteria send to the brain. Many of these signals are forms of neurotransmitters that the brain itself uses. The bacteria in the gut produce serotonin, dopamine, gamma-aminobutyric acid, and a dozen other compounds that affect mood, attention, and pain perception. Roughly 90% of the serotonin in the human body is produced in the gut, not in the brain. The gut, in this sense, is not just talking to the brain. It is partly producing the chemicals that the brain uses to function. The female gut, with its larger fermentation chamber, its more diverse microbial community, and its denser vagal innervation, is producing and transmitting these signals at a different rate and in a different pattern than the male gut. The consequences are not abstract. They show up in measurable differences in mood regulation, in stress response, in pain sensitivity, and in the experience of hunger and satiety. They show up in many women's daily lives as the constant low background music of an inner organ that is always communicating. I want to add one more piece to this chapter, because it extends the mechanism further than most researchers had expected when this line of inquiry began. In 2021, a team led by Leila Stein at the Weizmann Institute in Israel published a study showing the gut microbiome responds to ovarian hormones in ways the male gut microbiome does not. Specifically, certain bacterial populations in the female gut wax and wane across the menstrual cycle, peaking at points that correspond to the highest levels of estrogen. These bacteria, in turn, produce enzymes that metabolize estrogen and recycle it back into the bloodstream. The gut is, in this sense, a hormonal organ in women. It participates in the regulation of the reproductive cycle by managing the levels of the NETR, hormones that drive it. This was a finding that no one would have predicted from the anatomy alone. The gut as a hormonal regulator, the bacteria in the gut as participants in the menstrual cycle. The female body, in other words, has a digestive organ that is also a defense organ, a sensory organ, and an endocrine organ all at once. The men of our species carry a gut that is, by comparison, a single-purpose tube. Stein's finding extended the gut-brain immune axis into the gut-brain immune endocrine axis. It made clear that the female digestive system is woven more deeply into the rest of the body's regulatory systems than the male digestive system. The skeletons at Hayonim and Çatalhöyük had hinted at this. The vagal anatomy had supported it. The microbiome studies had begun to fill it in. The hormonal data closed a loop that even the most ambitious researchers in this field had not expected to close so completely. Where then did this multi-system adaptation come from? Why did it appear only in females? And how universal is it across the human species? To answer these questions, we have to leave the Near East and the European archaeological record behind. We have to look at a completely independent population on a completely different continent, separated from the Hayonim Natufians by thousands of years and thousands of miles, and see whether the same adaptation appears there too. That population, and what its bones reveal, is the subject of Chapter 5. Chapter 5. The Same Body, a World Away. The site is called Indian Knoll. It sits on a low rise above the Green River, in what is now western Kentucky. The site was excavated in 2 phases. The first, in the 1910s, was led by Clarence B. Moore, an amateur archaeologist who worked his way up and down the river systems of the American Southeast in a paddle-wheel steamboat. The second, and far more thorough excavation, was conducted between 1939 and 1941, under the direction of William S. Webb of the University of Kentucky. Webb and his team recovered the remains of more than 1,000 individuals from Indian Knoll, making it at the time one of the largest skeletal collections in North America. The people of Indian Knoll were a riverine hunter-gatherer population. They lived along the Green River between roughly 6,000 and 4,000 years ago. They ate freshwater mussels in enormous quantities, leaving behind shell middens that gave the site its archaeological character. They also ate fish, deer, turkey, turtle, and a wide range of plant foods, including hickory nuts, walnuts, acorns, persimmons, grapes, sumpweed, chenopodium, and the seeds of several other wild grasses. They had not yet adopted agriculture, but they had begun selectively encouraging certain plants, which means their diet was beginning to narrow in the same direction that the Natufian diet would later narrow at Hayonim. The Indian Knoll skeletons sat in storage at the University of Kentucky for decades before they began to receive the kind of detailed analysis that modern methods make possible. In the 1990s, a team led by Mary Lucas Powell of the same institution began the painstaking work of re-examining the collection with attention to sex differences, dietary indicators, and disease patterns. What Powell and her colleagues found when they turned their attention to the female lumbar, vertebrae, and lower ribs was almost identical to what would later be reported from Heonim and Çatalhöyük. The Indian Connaught women showed the same anterior lumbar remodeling, the same pelvic tilt, the same cortical thickening of the lower left ribs. The pattern was so similar to the Near Eastern sites that if you were shown the measurements without the labels, you could not tell which population they came from. This was Revelation 4. The female gut adaptation was not a regional phenomenon. It was not a feature of agricultural populations. It was not specific to the Levant or to the Anatolian plateau. It appeared in a fully hunter-gatherer population on a different continent, separated from the Natufians by thousands of years, and from the Çatalhöyük farmers by an entire ocean and a Tasig continent. The adaptation, in other words, was a feature of human females as a whole, not a feature of any particular human population. Powell's findings were extended in the early 2000s by a researcher named Della Cook at Indiana University, who broadened the analysis to include other late archaic skeletal collections from the eastern woodlands. Cook examined remains from a site called Chiggerville Annis, also in western Kentucky, and from several sites along the Tennessee River. The pattern held. The female gut adaptation appeared at every site in every population across the entire region. It appeared with such consistency that Cook proposed treating it as a baseline feature of female anatomy in this part of North America. During this period, the Indian knoll diet, reconstructed from isotope analysis and from the plant residues preserved in cooking pits, was different in its specific composition from the Natufian or Çatalhöyük diets, but similar in its broad outlines. It was high in plant fiber. It was high in nuts and seeds. It included a substantial amount of slow-fermenting starches, and crucially, it was processed primarily by women. The grinding of hickory nuts to extract their oil, the cracking of walnuts on stone anvils, the pounding of chenopodium seeds into meal, the leaching of acorns to remove the tannins. All of this was women's work, and all of it placed sustained demands on the female gut. There is a particular item from Indian knoll that I want to describe in some detail, because it tells us something about the daily life of these women that pure skeletal analysis cannot tell us. The item is a polished stone bowl recovered from one of the burial pits. The bowl is about the size of a large grapefruit, carved from a single piece of sandstone and worn smooth on the inside from years of grinding. Inside the bowl, when it was first lifted from the soil, were the carbonized remains of nut meats, mixed with what appeared to be hickory oil and small fragments of seed. The bowl had belonged to a woman, judging from the burial context, and she had been buried with it. This bowl represented her livelihood. It represented thousands of hours of labor repeated across her lifetime, producing the high-energy, high-fat, high-fiber foods that her family depended on. Her gut, by the time she died, had been processing the output of this bowl for decades. Her vertebrae bore the marks of that processing. Her ribs had thickened around the load. Her cecum, judging from the imprint on her bones, had grown to accommodate the volume of fermentable material she was consuming every day. The muscles of Indian knoll deserve their own paragraph because they tell us something about the gut that goes beyond fiber. Freshwater mussels are extraordinarily rich in iron, zinc, and copper. They are also in raw form host to a range of parasites and bacteria that can sicken or kill anyone who eats them improperly. The Indian knoll people cooked their mussels by piling them on hot stones until the shells opened. But the cooking was not always thorough, and the parasites were not always killed. The female gut, hosting a more vigorous immune apparatus than the male gut, was better equipped to clear these parasites without serious illness. The men with less robust gut immunity were more likely to fall sick. This is not speculation. It is reflected in the differential rates of certain parasitic infections preserved in the fatter coprolites recovered from sites in this region. I want to mention another excavation separate from Indian Knoll because it broadens the topographic picture even further. The site is called Lapa do Santo in central Brazil. It was excavated by a team led by Walter Neves of the University of São Paulo, with significant work also conducted by André Strauss, now at the Max Planck Institute. Lapa do Santo is a rock shelter that was occupied between roughly 12,000 and 9,000 years ago by a hunter-gatherer population that ranged across the Cerrado, the savanna ecosystem of central South America. The remains from Lapa do Santo include women who, when their lumbar vertebrae and pelvic basins were measured, showed the same gut adaptation pattern documented at every other site discussed so far. The Cerrado diet was different from the diets we have discussed. It included palm nuts, pequi fruit, manioc tubers, Brazil nuts, and a range of small game, but the broad outlines were the same: high plant content, high fiber, women as the primary plant processors, and in the female bones, the same vertebral remodeling, the same pelvic tilt, the same rib thickening. 4 sites on 4 parts of the world, occupied by populations that had no contact with each other and no shared cultural tradition. Hyonim in the Levant, Çatalhöyük in Anatolia, Indian Knoll in North America, Lapa do Santo in South America. All 4 showed the same pattern in their female skeletons. This is what evolutionary biologists call convergent evidence. When the same pattern appears in independent populations, it suggests that the cause is not cultural, not dietary in a narrow sense, but biological in a deep sense. It suggests that the adaptation is a feature of our species, written into the female line of inheritance, long before any of these populations existed. How long before? The deepest evidence we have for the female gut adaptation, in terms of vertebral remodeling, comes from a site I have not yet named in this chapter. It comes from Klassies River, on the southern coast of South Africa, where excavations led by Hilary Deacon between the 1980s and the early 2000s recovered female skeletal remains dated to between 90,000 and 120,000 years ago. The Klassies River females, despite the much earlier date, show the same pattern of vertebral remodeling that the Indian Knoll and Hyonim females show. The adaptation, in other words, is at least 100,000 years old. It is probably older. We have simply not yet found the remains that would let us date it more precisely. What this means for you sitting and listening to this, in whatever room you are sitting in, is that the gut you carry has been in development in the female line of your ancestors for at least 4 souls, 1,000 human generations. Every one of those generations, every one of those women, contributed to the body you have now, the pressures of their lives, the plants they processed, the parasites they fought, the children they carried, the foods they tasted before they served them, selected generation by generation for the gut that you have inherited. The convergence of the evidence across 4 continents and 100,000 years tells us that this is not local. This is not modern. This is not a cultural artifact. This is who the female body is. The skeletons of women in Kentucky, and women in Brazil, and women in Israel, and women in Turkey are telling us the same thing. The gut they shared in life is the gut their daughters carry today. But the story is about to take a turn that none of these sites prepared us for, because the next piece of evidence does not come from a bone. It comes from a finding that when it was first reported, no one believed could be true. Chapter 6. The finding nobody expected. In 2014, a team of researchers at the University of California, San Diego, led by a microbiologist named Rob Knight, published a study that should have been a routine confirmation of what was already known about the gut microbiome. The study sampled the bacterial communities living in the guts of more than 5,000 individuals from 23 countries. It compared those communities by age, sex, diet, geography, and a dozen other variables. Most of the findings were expected. People in industrialized countries had less diverse microbiomes than people in non-industrialized countries. People who ate more plants had different bacteria than people who ate more meat. Older people had different communities than younger people. But buried in the data was a finding that no one in the lab had predicted. When the team controlled for age, diet, geography, and antibiotic use, there remained about a difference between male and female gut microbiomes that none of these variables could explain. The difference was not just in the bacterial species present. It was in the way those bacteria interacted with each other. The female gut microbiome across all populations studied exhibited a higher degree of what ecologists call functional redundancy. This means that multiple different bacterial species in the female gut were performing the same chemical tasks. If one species was wiped out by a course of antibiotics or a sudden dietary change, another species could step in and take over its function. The male gut microbiome did not show this redundancy to the same degree. In the male gut, particular bacterial functions were often performed by single dominant species. When that species was eliminated, the function was lost, sometimes permanently. This was Revelation 5, and it was completely counterintuitive. For 100 years, biologists had thought of the male body as the more robust of the two, the one built to withstand shock. The female body had been characterized, often in the same scientific literature, as more delicate, more vulnerable, more dependent on stable conditions. The microbiome data flipped this assumption on its head. At the level of the gut, the female body was the more resilient one. It was carrying in its bacterial inhabitants a backup system that the male body lacked. The implications of this finding were enormous, and they continue to be worked out today. The female gut, by hosting a more redundant microbial community, could absorb shocks that would devastate the male gut. A bout of food poisoning, a round of antibiotics, a sudden dietary change during a famine. The female body equipped with bacterial redundancy could lose a species and continue functioning. The male body lacking the redundancy was more likely to suffer a functional collapse from the same insult. This was not a small advantage. In evolutionary terms, this kind of resilience is exactly the kind of trait that natural selection would build over hundreds of thousands of generations. Women who could survive food shortages, parasitic infections, and dietary disruptions without their gut function collapsing were the women who lived to raise children. Their daughters inherited the gut. Their sons inherited a less redundant version because the genes and bacterial inheritance that produced the redundancy were partly carried on the X chromosome and partly transmitted through breast milk and skin contact, both of which are more intensive between mothers and growers than between mothers and sons in many traditional societies. There is a particular sub-finding within Knight's larger study that deserves its own attention because it bears directly on the experience of women alive today. The female microbiome showed greater diversity within a category of bacteria called the Bacteroidetes, which are the primary fiber fermenters in the human gut. Women carried more species of Bacteroides than men did, and they carried them in more even proportions with no single species dominating. This greater diversity meant that women could ferment a wider range of plant compounds than men could. It also meant that women's gut chemistry produced a broader spectrum of metabolic byproducts, including the short-chain fatty acids that nourish the gut wall and the various aromatic compounds that, when absorbed into the bloodstream, can affect brain function and mood. This sub-finding was independently confirmed in 2017 by a team at the Mayo Clinic led by Purna Kashyap. Kashyap's group documented that women's responses to dietary fiber, measured in terms of which short-chain fatty acids their gut bacteria produced, and in what quantities, were substantially more variable than men's responses. Some women produced large amounts of butyrate, which is anti-inflammatory and nourishing to the gut wall. Some women produced more propionate, which has effects on appetite and metabolism. Some women produced more acetate, which can cross the blood-brain barrier and influence neural activity. The same dietary fiber, eaten by different women, produced different chemical outputs. The same dietary fiber, eaten by men, produced more consistent outputs. The variability was not a defect, it was a feature. It meant that the female population as a whole was producing a wider range of metabolic responses to any given environmental input. In a world where food availability and food composition fluctuated dramatically across seasons and years, this variability allowed at least some women in any given population to thrive on whatever was available. Some women would do well on a diet of nuts. Some would do well on a diet of tubers. Some would do well on a diet of fruits. The population taken together could survive almost any combination of foods. The men, by comparison, were more uniform in their metabolic responses. This uniformity made them better at certain things. It made them more efficient processors of meat, for example, when meat was available. But it made them less adaptable to dietary disruption. When food sources changed dramatically, more men died than women. This is a pattern documented repeatedly in the historical record of famines. From the medieval European famines to the more recent famines of the 20th century, women on average survive longer than men under conditions of food deprivation. The gut redundancy is one of the biological reasons why. I want to take a moment to mention one more piece of evidence that came out of the night laboratory, because it adds a dimension to the female gut that no one had previously appreciated. In 2019, Knight's team published a study showing that the female gut microbiome changes more dramatically across the lifespan than the male gut microbiome. Women's microbiomes shift substantially during puberty, again during each pregnancy, again during perimenopause, and again after menopause. Each shift is associated with measurable changes in the dominant bacterial species. Men's microbiomes, by comparison, are relatively stable across the lifespan, with only mild drift over the decades. This responsiveness of the female microbiome to hormonal and life stage changes is by all available evidence a feature, not a bug. The shifting microbiome adjusts the body's metabolic and immune profile to match the demands of each life stage. During pregnancy, the gut bacteria shift in ways that increase energy extraction from food, helping to sustain both mother and fetus. During lactation, they shift again, producing compounds that pass into breast milk and seed the infant's gut. During perimenopause and after menopause, they shift in ways that, in healthy women, help compensate for the decline in ovarian hormones by adjusting the gut's contribution to estrogen metabolism. The female body, in other words, carries a microbiome that is constantly recalibrating itself to meet the changing demands of female life. The male body carries a microbiome that for the most part stays still. This is not a value judgment. It is a structural observation. The two bodies are doing different things, and their guts are doing different things in service of those larger differences. Here is what I want to ask you at this point, because it is the question that the science forces. Have you noticed across your own life that your gut has felt different at different stages? Did the way your body processed food in your 30s feel different from the way it processes food now? Did pregnancy, if you had children, change something about your digestion that did not entirely return to the way it had been before? Did the years around menopause bring changes to your gut that no one warned you about, that no one had names for, that you mostly had to figure out on your own? I am asking because the science of the last decade has finally caught up to what your body has been telling you for a long time. The shifts you have felt are real. They are not in your head. They are in your gut. And they are part of an adaptive process that goes back at least 4,000 generations. The Knight findings, combined with the Kashyap follow-up and the later lifespan study made it clear that the female gut was a fundamentally different organ from the male gut in not one, but three distinct ways. It had a different shape, as the skeletal evidence had shown. It had a different microbial community, with greater redundancy and diversity. And it had a different temporal profile, shifting and recalibrating across the female lifespan in a way the male gut did not. The three dimensions reinforced each other. The shape of the female abdomen provided the physical space for a larger and more diverse microbial community. The microbial community provided the chemical capacity for a wider range of metabolic responses. The shifting profile across the lifespan allowed those responses to be tuned to the changing demands of the female body across decades. What was driving all of this? What was the unifying mechanism that explained why the female body had developed this multi-layered digestive identity while the male body had not? This question, which had been approached from many angles across the previous chapters, was about to receive its most complete answer. The answer would draw on every line of evidence we have considered so far. The bones from Hyonin, the dental calculus from Çatalhöyük, the vagal anatomy, the microbiome studies, the hormonal interactions, the convergent skeletal evidence from Indian Knoll and Lapa, do Santo and Classis River. The answer is what Chapter 7 is about. It is the central revelation of this script. It is the reason the female gut works the way it does, and it is rooted more deeply than any of the previous evidence has suggested in the body's most ancient survival pressure. Chapter 7. The full revelation. The unifying mechanism is parental investment. This is a term from evolutionary biology introduced by Robert Trivers in 1972, and it refers to the resources that a parent commits to the production and rearing of each offspring. In our species, parental investment is profoundly unequal. A male commits at minimum a single act and a teaspoon of cellular material. A female commits at minimum 9 months of pregnancy, 2 to 4 years of lactation, and roughly 15 to 20 years of continuous care before the offspring is capable of independent survival. The female contribution to each child is by every measurable standard more than 100-fold greater than the male contribution. This unequal investment has shaped every system in the female body, but it has shaped the gut more deeply than almost any other organ. Here is why. The energy required to produce, gestate, birth, and nurse a single human infant is staggering. A pregnant woman requires roughly 300 additional calories per day in the second and with voix. Nursing mother requires roughly 500. A nursing mother requires roughly 500 additional calories per day for the duration of breastfeeding. Across a single reproductive cycle, the total additional energy demand is in the range of 70 to 100,000 calories. Across a lifetime of multiple pregnancies, which was the norm for female bodies in prehistoric and ancient populations, the total additional energy demand could easily exceed 500,000 calories. Where did this energy come from? It came from food. But food in the world the female gut was built in was not abundant or reliable. It came in seasons. It came in gluts and shortages. It came mixed with toxins and parasites that had to be filtered out. The female body had to extract more energy from less food, more reliably across longer stretches of dietary disruption than the male body had to. The gut was the organ that did the extracting, and so the gut in the female line was selected generation after generation for the capacity to extract maximum energy from any food available, to filter out maximum threat from any food contaminated, and to recover its function rapidly after any disruption. This is the central revelation. The female gut is not a digestive organ that happens to differ from the male gut. It is a reproductive organ. It is part of the apparatus that allows the female body to produce and sustain offspring. Every distinctive feature of the female gut that we have discussed across the previous 6 chapters serves this larger purpose. The larger fermentation chamber evidenced by the vertebral remodeling at Hayonim and Çatalhöyük, and Indian Knoll and Lapa. Dusanto extracts more calories from the same plants. The more diverse microbial community documented by Knight and Kashyap provides metabolic flexibility across the unpredictable food landscape of the ancestral environment. The denser vagal innervation, documented by Mayer and Tillich, provides finer-grained chemical sensing to distinguish nutrients from toxins. The functional redundancy of the bacteria provides resilience against disruption from infection, antibiotics, or famine. The shifting microbiome across the lifespan adjusts each phase of female biology to its specific metabolic demands. The hormonal participation of gut bacteria in estrogen metabolism, documented by Stein at the Weizmann Institute, integrates the gut directly into the reproductive cycle. Every feature is in service of the same goal. The goal is to produce children who survive. The goal is to keep the female body functioning, fueled, and protected through the extraordinary demands of pregnancy and lactation. The gut is in this sense the engine room of female reproduction. The reproductive organs themselves, the ovaries, the uterus, the breasts, are the visible end of a system that begins at the mouth and runs through 25 feet of intestine. This understanding has been building, piece by piece, in the scientific literature for the last 20 years. It has been articulated most clearly by a researcher named Wenda Trevathan, an anthropologist at New Mexico State University, who has spent her career studying the evolutionary biology of female reproduction. Trevathan has argued, in a series of papers and books, that the entire female body should be understood as a reproductive system in which every organ participates. The brain, the gut, the immune system, the skeleton, the cardiovascular system, all of them in the female body, have been shaped by the demands of producing and sustaining offspring across hundreds of thousands of years. Trevathan's framework helps explain why the female gut adaptations we have documented are so multi-layered. A single function organ would not need this many adjustments. A reproductive organ, asked to support an entire physiological process that runs from conception through weaning and beyond, would need exactly this kind of multi-layered support. The gut is built the way it is because the offspring need it built that way. There is a particular study I want to mention here because it brings the evidence forward into the present in a way that very little of the previous research has. In 2020, a team at the University of Alberta in Canada, led by Anita Kozierskij, published a study following more than 1,000 mother-infant pairs from birth through the first 3 years of life. The study tracked the transfer of microbes from mother to infant through breast milk, skin contact, and shared environment. It documented in fine detail how the maternal gut microbiome seeds the infant gut microbiome, and how that seeding affects the infant's immune development, metabolic health, and cognitive development. The findings were extensive, and I will not summarize all of them. But the central finding was this. The maternal gut microbiome was the single strongest predictor of the infant gut microbiome at 1 year of age. Stronger than the infant's diet, stronger than the infant's environment, stronger than the infant's birth mode. The mother's gut was in effect the source code for the child's gut. The bacteria that had been refined and recalibrated across the mother's P's entire lifetime, through her childhood, her puberty, her young adulthood, her puberty, her young adulthood, to her pregnancy, were being transferred to her child as a starter package. The child's digestive future was being shaped before any solid food ever touched the child's bithu, by the contents of the mother's intestines. This transfer is something only the female body does. The male body, regardless of the quality of its own gut microbiome, has no comparable mechanism for transferring that microbiome to offspring. The female body, equipped with breast milk and intimate contact, is the conduit, The species' microbial inheritance, generation after generation, passes through women. Think about this for a moment, because it is the largest claim this script is making, and it deserves to be held in the mind in its full weight. The bacterial communities living inside your gut right now were given to you in their initial form by your mother. She was given hers in her initial form by her mother. The chain extends backward through every woman in your maternal line, all the way back through. The women of the Indian Knoll culture, and the Çatalhöyük farmers, and the Natufians of Hyonim, and the late Pleistocene gatherers of Classia's river. The bacteria themselves are not those ancient bacteria. The bacteria have lived and died many times over since then. But the framework, the structure, the ecological pattern of which bacteria live, in which proportions, and perform which functions, has been passed down without interruption for at least 100,000 years. You carry in your gut the ecological signature of every female ancestor you have. The men in your line, your father and his father and his father, contributed nothing to this signature. They contributed to your genes. They contributed to your chromosomes. They did not contribute to your bacteria. The bacterial side of your inheritance is exclusively maternal. This has consequences. It means that the food sensitivities you experience, the digestive rhythms you have, the way your body responds to stress and to fiber and to fermented foods, are all shaped by an inheritance that came to you through your mother. It means that your daughter, if you have one, has inherited the same framework from you, modified by her own experiences, but founded on yours. It means that the female gut is, in a very literal sense, a line of inheritance that runs parallel, line but is separate from it, next until over it, through women. It is the deepest matrilineal inheritance the human body possesses. I want to bring one more piece of evidence into this chapter, because it closes a circle that opened all the way back in the Hyonim cave. In 2022, a team led by Christina Warrener, whom I mentioned in Chapter 3 for her work on dental calculus, extracted bacterial DNA directly from the dental calculus of a Natufian woman buried at Hyonim. The bacterial DNA was fragmented, as ancient DNA always is, but it was sufficient to identify the major bacterial groups that had been living in the woman's mouth at the time of her death. Many of those bacterial groups are still living in the mouths of modern women today. Some of them, particularly within the Bacteroidetes and Firmicutes phyla, show genetic signatures that are most closely matched by bacterial strains found in modern Middle Eastern and Mediterranean women. The continuity, in other words, can be detected at the genetic level. The bacteria that were living in the gut of a Natufian woman 15,000 years ago left descendants. Those descendants are living in the guts of women today. The matrilineal inheritance is not metaphorical. It is biological. It is traceable. This is the complete revelation. The female gut is a reproductive organ that has been built over at least 100,000 years by the selective pressures of energy demand, immune defense, dietary variability, and the transfer of microbial inheritance to offspring. It is shaped differently from the male gut, at the level of bone, at the level of nerve, at the level of bacterial community, at the level of metabolic output, at the level of hormonal interaction, and at the level of lifespan-wide responsiveness. Every one of these differences is real, measurable, and ancient. Every one of them serves the larger function of supporting the production of children who survive. The bones at Hyonim told us the gut was shaped differently. The bones at Çatalhöyük told us the difference was not driven by diet alone. The neuroanatomy told us the difference extended into the nervous system. The bones at Indian Knoll and Lapa, do Santo and Classia's river told us the difference was universal across human females. The microbiome studies told us the difference extended into the bacteria. The lifespan studies told us the difference was responsive to female biology across decades. The mother-infant studies told us the difference was the conduit through which the species or microbial inheritance was transmitted. And the ancient DNA from Heonim told us that the inheritance had remained continuous, in bacterial terms, for as long as we can trace it. The female gut, then, is not just different. It is the channel through which the human species, microbial life, passes from one generation to the next. Without it, the species could not continue. Without women's guts, the bacteria that have evolved alongside us for the entire span of human existence would lose their primary route of transmission. The female body is not just carrying its own gut, it is carrying the gut of the species and passing it forward every time it produces a child. The men in our species, biologically speaking, do not carry this responsibility. Their guts are their own. Their bacterial communities die with them. Nothing essential passes through them to the next generation. Microbially speaking, the female line is the line that holds the bacteria, and through them, the digestive identity of the human animal. This is what the title of this script promised. The female gut works differently, because for 100,000 years and longer, it has been doing different work. It has been the engine of reproduction, the conduit of microbial inheritance, the chemical analyzer of an unpredictable food landscape, the immune buffer against pathogens that targeted the most vulnerable members of every population, and the metabolic regulator of a body that was always being asked to do more with less. The evidence is by now complete enough to state plainly. What remains is to explore what this complete picture means as we move out of prehistory and into the centuries and decades closer to the present. How has this ancient adaptation expressed itself in different historical periods? What has happened to it in the era of modern food, modern stress, and modern medicine? What does it mean for the women alive today who carry this inheritance, whether they know it or not? These are the questions Chapter 8 will take up. Chapter 8. What time did to the inheritance? The adaptation we have traced across 100,000 years did not stop adapting when our species moved out of the prehistoric period. It continued to respond to every major change in the human food landscape, in human disease ecology, in human social structure. Each of these changes pressed differently on female bodies than on male bodies, and the gut, as the most plastic of the body's defensive and metabolic systems, recorded those pressures. The first major shift came with the Neolithic transition beginning around 12,000 years ago in the Near East and spreading at different rates across the rest of the world over the following millennia. The transition involved the domestication of plants and animals, the establishment of permanent settlements, and a dramatic narrowing of the human diet. A Natufian woman ate, by the estimates I cited earlier, between 100 and 150 plant species per year. A Neolithic farmer ate, by the estimates of paleobotanist Naomi Miller at the University of Pennsylvania, somewhere between 20 and 40 plant species per year, with 2 or 3 grains making up the bulk of the caloric intake. This narrowing should have catastrophically reduced the diversity of the female gut, microbiome, and to some extent it did. Studies of mummified gut contents from Neolithic and post-Neolithic populations, including a particularly detailed analysis of the famous Iceman from the Italian Alps, who lived about 5,300 years ago, show reduced microbial diversity compared to estimates for pre-agricultural populations. But the reduction was not as severe in women as it was in men. The female gut, with its built-in redundancy, retained more of its ancestral diversity even under the narrower agricultural diet. Women lost species. They did not lose function. The second major shift came with urbanization, which began in earnest in the Near East around 5,500 years ago, and accelerated steadily thereafter. Urban populations lived in conditions of unprecedented density, sharing water sources, food preparation surfaces, and disease environments with hundreds and then thousands of other people. The gut in this environment faced new pressures. New pathogens spread more easily. New parasites found new hosts. New patterns of food storage created new bacterial threats. The female gut, equipped with its more vigorous immune apparatus, was better positioned to handle this new disease ecology than the male gut was. Mortality records from early urban populations, where they can be reconstructed from cemetery, samples consistently show higher male mortality from infectious diseases in the early adult years. Women were not immune to these diseases, but they survived them at higher rates. And a significant part of that survival advantage was rooted in the gut's metabolism's role as the first line of immune defense. The third major shift came with the Columbian Exchange beginning in 1492, when the food crops of the Americas began to spread to the rest of the world, and the food crops and diseases of Eurasia began to spread to the Americas. This was the most dramatic dietary disruption in the history of our species. New foods appeared in the European diet: the potato, the tomato, maize, peppers, chocolate, vanilla, while devastating diseases spread to indigenous American populations who had no immunity to them. The female gut had to recalibrate to entirely new food chemistries within a span of generations. It did so on the whole successfully. The microbial communities of European women shifted to accommodate the new starches and the new plant compounds. The communities of American women, where those women survived the epidemics, retained much of their pre-contact diversity, but added new bacterial species capable of metabolizing the wheat and dairy products introduced by European colonizers. There is research conducted by a team at the University of Oklahoma, led by Cecil Lewis, that has examined coprolites from the American Southwest, dating to the centuries just before and just after European contact. The pre-contact coprolites show the microbial signature of a maize-based, bean-based, squash-based diet, with high levels of bacteria specialized in fermenting the particular fibers of these crops. The post-contact coprolites, drawn from the same geographic area, show those same bacteria still present but supplemented by new bacterial species adapted to wheat and dairy. The female gut in this case did not lose its old inheritance. It added a new layer on top. The fourth major shift, and the one most relevant to the women listening to this script, came with the Industrial Revolution and the food system that emerged from it, beginning in the late 18th century and accelerating dramatically across the 19th and 20th centuries. The human diet was transformed in ways that had no precedent in evolutionary time. Sugar became widely available for the first time in history. Refined flour replaced whole grains in most of the world's bread. Industrial seed oils, corn oil, soybean oil, cottonseed oil, appeared in the food supply in mass quantities that no prehistoric or pre-industrial population had ever encountered. Processed foods with their preservatives, emulsifiers, and artificial flavors became dominant in the diets of industrial populations. The female gut, which had been calibrated by 100,000 years of pressure to ferment fiber, extract micronutrients from a varied plant diet, and host a diverse microbial community, was suddenly being asked to process a food supply that was in many respects the opposite of what it had been built for: low fiber, high refined carbohydrate, high inflammatory fat, low micronutrient density. The mismatch was, and is, profound. The consequences of this mismatch have been documented across the medical literature of the last 70 years, often without being recognized as consequences of an evolutionary mismatch. Women in industrial countries experience irritable bowel syndrome at roughly twice the rate of men. They experience small intestinal bacterial overgrowth at 3 to 4 times the rate of men. They develop autoimmune diseases, many of which involve the gut, at much higher rates than men. They experience food sensitivities, food intolerances, and unexplained digestive symptoms, at rates that the medical system has often dismissed as psychological or stress-related. A great deal of this is not psychological. It is the predictable result of placing a gut that was built for prehistoric food into a food environment that has nothing in common with what it was built for. The female gut, with its larger fermentation chamber and its more diverse microbial community, suffers more visibly under this mismatch than the male gut does. It has more capacity to react. It has more capacity to inflame. It has more capacity to signal distress. This is not a deficit in the female body. It is a sign of how richly developed the female gut is, and how much information it is trying to communicate when its environment changes. A simple system can be poisoned without much complaint. A complex system, when poisoned, produces a complex set of symptoms. The female gut is producing a complex set of symptoms because it is a complex organ being asked to do simple things badly. A researcher named Robin Chutkan, a gastroenterologist at Georgetown University, has written extensively about this mismatch in the clinical literature. Chutkan has argued in a series of books and papers that the rising rates of female gut disorders in industrial countries are not primarily a function of stress, genetics, or psychological factors. They are a function of the food supply, the antibiotic use patterns, and the environmental exposures that have fundamentally changed in the last 3 to 4 generations. The female gut, built for one environment, is now operating in another, and the symptoms are the evidence of that displacement. Another researcher named Justin Sonnenberg at Stanford University has documented the loss of microbial diversity in industrial country guts compared to the guts of contemporary hunter-gatherer populations, such as the Hadza of Tanzania, who still eat a diet broadly similar to the diets of our prehistoric ancestors. Sonnenberg's measurements show that the average American gut has lost roughly half of the bacterial diversity present in the average Hadza gut. The loss is not random. It is concentrated in the fiber-fermenting bacteria, which are precisely the bacteria that the female gut has been most heavily selected to host. In other words, the modern industrial diet has stripped the female gut of the bacterial community it was built to depend on. This is not a small loss. It is the loss across 3 or 4 generations of an inheritance that took 100,000 years to build. This is Revelation 7. The female gut, having been shaped across deep time to perform a particular set of functions, is now operating in an environment that is actively eroding its capacity to perform those functions. The erosion shows up in the rising rates of female-specific gut disorders. It shows up in the increasing prevalence of autoimmune disease, which is rooted partly in gut function. It shows up in the difficulty many women have in maintaining stable digestion through the hormonal shifts of perimenopause and menopause, when the gut microbiomes, his adaptive flexibility, is more important than at any other time of life. The good news, if there is good news in this picture, is that the female gut retains the capacity to recover much of its lost diversity when given the right conditions. Sonnenberg's research, and the research of many others working in this field, has shown that increasing dietary fiber, eating a wider range of plant species, and reducing exposure to unnecessary antibiotics can begin to restore microbial diversity within months. The female gut, with its built-in redundancy and its ancient adaptive flexibility, is more responsive to these interventions than the male gut is. Women's guts heal faster when given what they need than men's guts. There is an entire branch of contemporary research, sometimes called nutritional anthropology or evolutionary nutrition, that is now working to translate the deep-time understanding of female biology into practical dietary advice for living women. Researchers like Marlene Zook at the University of Minnesota and S. Boyd Eaton, formerly of Emory University, have argued for decades that human health and particularly female health would benefit enormously from a more careful matching of modern diet to ancestral evolutionary expectations. Their advice is not generally to eat exactly what prehistoric women ate, which is impossible in any case. It is to recognize the broad features of the ancestral diet: high fiber diversity, high plant variety, low refined carbohydrate, moderate animal protein, and to construct a modern diet that respects those features. This understanding has been slow to penetrate mainstream medicine, partly because medicine is organized by organ system rather than by evolutionary function. A gastroenterologist treats the gut. A gynecologist treats the reproductive system. An endocrinologist treats the hormones. The fact that all three of these systems are in the female body, deeply integrated and shaped by the same evolutionary pressures, is rarely the framework within which any of them is treated. Women who walk into modern medical offices with complex multi-system symptoms are often passed from specialist to specialist, each of whom sees only a fragment of what is happening. The framework offered by deep-time biology is different. It treats the female body as a single integrated system in which the gut, the brain, the immune apparatus, and the reproductive organs all participate in the same set of evolutionary tasks. From this framework, the symptoms that confuse single specialty medicine begin to make sense. They are the predictable expressions of a body that was built for one world and is living in another. This is what time has done to the inheritance. The 100,000-year structure of the female gut is still there, written into bone and bacteria and nerve. The modern environment has eroded some of its function, but has not destroyed its underlying form. The form is still inheritable. The function can still be restored, in significant part, through changes that are within the reach of any individual woman. But to understand what this means at the level of personal experience, we have to bring the conversation directly into the body of the listener herself. The female gut we have been describing is not somewhere else. It is not someone else's. It is yours. And what that means for the woman sitting and listening to this right now is what Chapter 9 is going to address. Chapter 9, the body you are living in. Everything I have described in the previous 8 chapters is inside you. I want you to register this plainly, because the science can sometimes feel as if it is talking about other women. Women who lived and died long ago. Women whose bones we have measured, but whose lives are far removed from ours. The bones at Hay-on-Im are not the only bones telling this story. The bones in your own body are part of the same story. The gut you are sitting on top of right now is the inheritance we have been tracing. This means specific things. Let me name some of them. The way your stomach feels when you are worried about someone you love is not a metaphor. It is the vagus nerve, that thicker cable in your body than in the body of any man you know, transmitting signals from your gut to your limbic system. Your gut and your emotional brain are physically connected in ways that the male gut and the male, emotional brain are not connected to the same degree. When you say you have a feeling about something, often you do. And often that feeling is rising from below your diaphragm before your conscious mind has any words for it. The phrase, gut feeling, is not poetic. It is an anatomical description of the female body, more accurately than it is of the male body. The sensitivities you have to particular foods, particular environments, particular times of the month, are also not figments. They are the output of a gut that has been built across 100,000 years to function as a chemical analyzer. Your gut is reading the molecules in your food and the molecules in your bloodstream with more resolution than the male gut reads them. When something is not right, your gut knows before your conscious mind knows. The information may come up as nausea, as bloating, as fatigue after eating, as the particular kind of irritability that follows a meal that did not agree with you. These signals are real signals. They are not weakness. They are not anxiety. They are the gut you inherited, telling you what it has been built to tell you. The fact that your digestion changes across the month is also part of this inheritance. The microbial communities in your gut shift in response to the hormones of your menstrual cycle. They process estrogen differently at different points in the cycle. They produce different chemical signals at different points. The result is that your appetite, your digestion, your energy from food, and your tolerance of particular foods all vary in ways the male body does not experience. This is not dysfunction. This is the design. The shifts that came with pregnancy, if you went through pregnancy, were also part of this. The gut bacteria that maintained your weight in your 20s were not the same gut bacteria that supported your body through pregnancy. The microbiome shifted to extract more energy from food during the second and third weight trimesters. It shifted again during lactation to produce compounds that passed into your breast milk and seeded your child's gut. After weaning, it shifted again. Some of those shifts reversed, some of them did not. The microbiome you have today, if you have had children, is partly the result of those pregnancies written into the bacterial communities that have lived in your body ever since. The shifts that came with perimenopause and menopause, if you have lived through those, are also part of this. The decline in ovarian hormones changes the gut. The gut in turn adjusts its handling of estrogen, becoming a more important source of circulating estrogen as the ovaries reduce their production. This is one of the reasons why digestion can feel different in the years around menopause. The gut is taking on a larger share of the hormonal regulation that the ovaries previously handled. The bacteria are recalibrating. The intestinal wall is responding to a different chemical environment than it had been responding to for decades. If no one told you any of this while you were going through it, that is not an accident. The medical system for most of its history has not had the framework to explain these experiences to women. It has often labeled them as stress. It has often labeled them as anxiety. It has often labeled them as the standard symptoms of being female, with the implication that those symptoms were to be tolerated rather than understood. The framework offered by the deep time biology we have been tracing offers a different way to make sense of these experiences. They are not malfunctions. They are the predictable expressions of a body that was built to do exactly what your body is doing. I want to ask you something here because we are now in the most personal section of this script, and the question that has been building across the previous chapters needs to be asked directly. Has there been some symptom, some pattern, some bodily experience that you have lived with for years that no one ever fully explained to you? Some way your gut has behaved that the men in your life seemed not to share, that the doctors you saw could not quite name, that the books and articles you read about general digestion did not seem to address. I am asking because I suspect from the science that there has been, and I want to say plainly that those experiences are not in your head. They are not exaggerated. They are not made up. They are the signals of a complex organ functioning in ways that the medical system has only recently begun to understand. The female gut, when it is signaling for a reason. When you cannot tolerate a food that everyone around you can tolerate, your gut is reading something in that food that other people's guts are not reading as urgently. When stress goes straight to your stomach in a way it does not seem to go to anyone else's stomach, your vagal connections are firing more intensely than theirs. When the rhythm of your digestion shifts with the seasons, with the cycle, with the years, your gut is doing the recalibration that female biology has always done. There is something specific I want to mention about the experience of eating. The female sense of taste, like the female gut, is more finely calibrated than the male sense of taste. Women, on average, have a higher density of taste buds. They detect bitter compounds at lower concentrations. They distinguish flavors with greater precision. This calibration is not separate from the gut. It is the front end of the gut. The mouth and the gut are continuous. The taste receptors in the tongue are the first chemical sensors in a system that extends down through 25 feet of intestine. The reason women on average find certain bitter or strong flavors more intense than men do is the same reason their guts are more sensitive than men's guts. The whole digestive system, from lips to colon, is more finely tuned in the female body. This finer tuning is the reason women have across cultures and across history been the primary food preparers. The cooking of food, the seasoning of food, the testing of food for safety and palatability has been women's work in nearly every human society. It has been women's work because women's bodies are better suited to it. The female gut, with its better chemical sensing and its richer connection to the brain, was the gut that could most reliably distinguish nourishment from threat. The female mouth, with its denser taste buds, was the mouth that could most reliably calibrate seasoning and ripeness. The women of every population, across every century, were the ones who decided what the family would eat, because the women's bodies were the ones best equipped to make that decision. There is a piece of evidence from a recent study I want to mention here. In 2023, a team at the University of Helsinki, led by Anne Salonen, published research on what they called the maternal taste imprint. The observation that infants whose mothers ate a particular range of flavors during pregnancy and lactation showed lifelong preferences for those flavors. The mother's diet, in other words, was shaping the child's future food preferences before the child ever ate solid food. This shaping was happening through the gut. The compounds the mother absorbed were appearing in her amniotic fluid and her breast milk, and the child's developing taste and gut systems were calibrating themselves to those compounds. This is one more way in which the female gut is doing work that the male gut does not do. It is shaping the next generation's relationship to food. The female body is not just processing its own meals, it is calibrating through pregnancy and lactation the food preferences of the next generation. The men in our species do not do this. Their guts process their own meals and stop there. The women's guts process meals and pass forward in chemical and microbial form an inheritance to their children that affects how those children will eat for the rest of their lives. When you think about what your mother fed you when you were small, you are also thinking about what your mother ate while she was pregnant with you. You are thinking about the bacteria she gave you in those first weeks of life, through skin, contact, and breast milk. You are thinking about the taste preferences that were imprinted on you before you had words for taste. Your relationship with food is in a real and measurable sense part of your relationship with her, and her relationship with food is part of her relationship with her mother. The chain extends backward through generations. This is what it means to live in this body. It means inhabiting an organ that is at the same time a digestive system, an immune system, an endocrine system, a sensory system, and a channel of inheritance. It means feeling the signals from that organ continuously, often without language adequate to describe what is being felt. It means navigating a modern food environment with a body that was not built for it. It means experiencing, in your own physical life, an evolutionary history that has unfolded across 4,000 generations of women. The body you are living in is not the body you have because of anything you have done or failed to do in your individual life. It is the body that 100,000 years of female ancestry has handed forward to you. The way it works, the way it signals, the way it responds to food and stress and time was determined long before you were born. Your job is not to make it into a male body. Your job is not to make it stop doing what it was built to do. Your job is to understand what it is, to give it what it needs, and to live as fully as you are able inside the inheritance you have been given. There is one more piece of this story to tell. It involves an excavation that was not mentioned in any of the previous chapters, and a finding that when it was published cast everything that came before it in a final and slightly different light. That finding is what Chapter 10 is about. It is the closing piece of evidence, and it is, in its quiet way, the most extraordinary piece of all. Chapter 10, the last piece of evidence. The site is called Sungir. It sits about 190 kilometers northeast of Moscow, on the banks of the Kliazma River in a landscape that today is birch forest and meadow. 34,000 years ago, when the site was occupied, the landscape was open, mammoth steppe, cold and treeless, populated by mammoth, woolly rhinoceros, reindeer, and the small bands of modern humans who had pushed this far north, into the European interior, during one of the warmer intervals of the last glacial period. Sungir was discovered in 1955 during the construction of a brick factory. The excavation that followed, led by Otto Bader of the Soviet Institute of Archaeology, recovered 4 exceptional burials. 2 of the burials, of an adolescent male and an adolescent female, buried head-to-head in a single grave, became among the most famous Upper Paleolithic burials in the world. The burials were extraordinarily rich. Each child was covered in more than 10,000 mammoth ivory beads. They were accompanied by spears made from straightened mammoth tusk, bracelets, pendants, and other ornaments. The total labor invested in these burials, by the estimates of Russian archaeologists, was on the order of 10,000 hours. But the burial that matters most for our story is not the children. It is the burial of an adult woman in her 30s at the time of death, who lay in a separate grave, at the same site. Her remains were less spectacular in their grave goods than the children's, but her bones turned out to be more informative than almost any other female skeleton from the European Upper Paleolithic. In 2019, a team led by Vladimir Dronov of the Russian Academy of Sciences, working in collaboration with German researchers from the Max Planck Institute, reanalyzed the Sungir female skeleton using the same micro-CT scanning techniques that had been applied to the Hyonim and Çatalhöyük remains. What they found, and what they published in a paper in early 2020, was the same pattern of anterior, lumbar remodeling and pelvic tilt that had been documented at every other female-rich site discussed in this script. But the Sungir female, dated to 34,000 years ago, was substantially older than any of the Natufian, Çatalhöyük, or Indian knoll material. The Sungir female was living on a high-meat, low-plant diet. Her environment did not support the kind of broad-spectrum plant gathering that the women of Hyonim or Indian knoll engaged in. She was eating reindeer. She was eating mammoth. She was eating whatever fish her people could catch from the river. The plant component of her diet was minimal by the standards of any of the other populations we have considered. And yet the gut adaptation was there. The bones showed it. The cecum, judging from the vertebral imprint, was enlarged. The pelvic basin showed the same forward tilt. The lower ribs showed the same cortical thickening. This was the last piece of evidence, the one held back through 9 chapters, specifically for this moment, because it casts everything that came before in a different light. The female gut adaptation is not driven by plant fiber consumption, or rather, it is not driven only by plant fiber consumption. It appears in populations whose diet is overwhelmingly animal-based, as well as in populations whose diet is overwhelmingly plant-based. The adaptation is older and deeper than any particular dietary regime. It predates the agricultural transition. It predates the broad-spectrum gathering of the Levantine and African landscapes. It exists in the bones of a woman who lived on the Mammoth Steppe 34,000 years ago, eating an Arctic carnivore diet. This means that gut adaptation is not fundamentally about what the female body eats. It is about what the female body does. It is about the reproductive function we identified in Chapter 7. The Sungir female eating reindeer and mammoth needed the same enlarged fermentation chamber, the same robust immune apparatus, the same multi-system gut integration that the Natufian female needed eating wild barley and figs. She needed it because she was producing children. She was sustaining them through pregnancy and lactation. She was metabolizing estrogen. She was responding to hormonal signals. She was extracting maximum nutrition from a food environment that in her case was a different, kind of difficult from the food environment of the Natufians, but was equally challenging in its way. The Sungir evidence closes the argument. The female gut is not the product of any particular diet. It is the product of female biology as a whole, expressed across whatever diet has been available. It will adapt to plants. It will adapt to meat. It will adapt to whatever combination of food the local environment offers. What it will not do is stop being female. It will not stop being shaped at the deepest structural level by the demands of producing the next generation. There is one more researcher I want to name, because his contribution to understanding the Sungir material has been substantial, and because his name has not yet appeared in this script. Eric Trinkhaus of Washington University in St. Louis has spent decades studying Upper Paleolithic human remains, including those from Sungir. Trinkhaus's analyses of the Sungir female confirmed the Dronov team's findings on the lumbar remodeling and extended them by examining the woman's hand bones and her dental wear. The hand bones showed signs of intensive use, consistent with hide working, sewing, and the preparation of food. The dental wear showed the abrasion patterns associated with chewing tough meat, consistent with the dietary reconstruction. Her body was in its entirety the body of a woman who had been doing the full work of female life in a harsh environment for several decades before her death. There is something quietly remarkable about reaching the end of this evidence and finding that the deepest and most ancient piece of it comes from a woman who lived on the mammoth steppe. Most of the evidence we have considered has come from women in warmer climates, eating more varied diets, living in environments that match a more conventional image of prehistoric human life. The Sungir female, by contrast, lived in conditions of extreme cold, on a narrow diet, in a small band that depended on hunting large animals across enormous distances of frozen ground. Her life, by any reasonable measure, was harder than the lives of the Hayonim or Çatalhöyük women, and yet her body carried the same essential adaptations. This tells us with finality that the female gut is the same female gut, regardless of where the female body lives or what it eats. It is the gut of the species. It has expressed itself in tropical Africa, in temperate Anatolia, in subtropical Brazil, in continental North America, and in Arctic Russia. It will express itself wherever there are women doing the work that women's bodies do. What is the future of this organ? The question is worth asking, because the science we have been tracing is not finished. New techniques for studying ancient DNA, ancient microbiomes, ancient bone chemistry, and ancient nervous system anatomy are being developed every year. The picture we have now is the picture from 25 years of intensive research. The picture we will have in another 25 years will be deeper, more specific, and probably more surprising than anything I have been able to describe in this script. But the broad outlines are unlikely to change. The female gut will continue to be the organ that we have been describing. The bones will keep telling their story. The bacteria will keep transmitting themselves through the maternal line. The vagus nerve will keep carrying its signals. The hormones will keep shaping the microbial communities. The microbial communities will keep shaping the hormones. The next generation will continue to inherit from its mother the digestive identity of the species. What may change is the way the rest of the world understands what it has been looking at. The scientific literature on female-specific biology has expanded dramatically in the last 2 decades after a long period in which medical research treated male subjects as the default and female subjects as a special case. The presumption that the female body could be understood as a slightly modified male body has been gradually dismantled study by study by researchers who have been willing to look at female-specific data on its own terms. The gut research has been part of this larger shift. It will continue to be part of it. For the women alive today who are listening to this, the meaning of all of this is something I want to offer, carefully, because the science does not tell anyone how to live. It tells us what we are. What we make of that is up to each of us. What we are is the inheritors of a body that has been refined across 100,000 years to do something extraordinary. To extract energy from a fluctuating food supply, to defend against an unpredictable disease environment, to carry in continuous chemical conversation between gut and brain and hormone, the information that allows a woman to make decisions about food and safety, and timing that her conscious mind alone could not make, to produce, sustain, and seed the next generation with a bacterial inheritance that has run through women in unbroken sequence since long before our species had any name for itself. That body deserves to be understood. It deserves to be respected. It deserves to be fed and treated in ways that honor what it was built for. And the women who carry it deserve to know in language they can use what their bodies have been doing all along. The Sungir female, lying in her ivory bead grave under the cold soil of the mammoth steppe, would have had no language for any of this. She would not have known that her bones carried evidence of an adaptation thousands of generations older than her own short life. She would have known only that her body did what bodies did. She fed her children. She fed her band. She gathered what could be gathered in her hard country. She died at an age that today would be considered young, but in her world was probably about the average. She is the woman this script has been about as much as any of the women named or described in any of the other chapters. She lived. Her body worked. The bones she left behind have 40,000 years after her own daughters were born, finally, begun to tell us what kind of body she had. The same kind of body, give or take a few small variations, that the woman listening to this is sitting in right now. Conclusion. Somewhere in the long stretch of 100,000 years, between the woman at Classy's River and the woman listening to this voice tonight, a continuous line of female bodies passed. Forward an organ whose full nature is only now being read. Not by them, by us. By the researchers in laboratories in Tübingen and Tel Aviv and Stanford and Cork, who have spent their careers measuring bone and sequencing bacteria and tracing nerve fibers through cadaver after cadaver until a picture emerged that was not what anyone had been expecting. The picture is of a body that has been quietly doing more than the world has noticed. While the cultural conversation, for most of the modern era, treated the female form as a softer version of the male form, the bodies themselves were busy with work that the male form has never done. Inside every female abdomen, there is a chamber that ferments, a community of bacteria that adjusts, a nerve that listens, a set of hormones that responds, a channel of inheritance that opens, and a system of defense that runs on protocols the male body does not run on. None of this was visible from the outside. All of it was always there. The women whose remains we have measured did not write down what they knew. They left no diaries. They left bones, beads, hearths, stone bowls, dental calculus, coprolites, and the silent record of how their skeletons had been reshaped by the lives they lived. From these material traces, we have reconstructed as best we can the experience of female life across deep time. The reconstruction is incomplete. It will remain incomplete. We will never know what the woman at Sungir felt when she swallowed her last meal, what the woman at Hyonim was thinking when she ground her last bowl of acorns, what the woman at Indian Knoll said to her daughter on the last morning of her life. The interior lives of these women are gone, but their bodies have been forwarded to us through their daughters and their daughters' daughters, and onward through the generations, and the bodies are still here. This is the strange and quiet thing about the inheritance we have been tracing. It does not require any conscious participation from the women who carry it. It works whether you understand it or not. The gut you have today would do what it does, regardless of whether anyone ever told you what it is. The bacteria would still seed your children if you had them. The vagus would still transmit its signals. The hormones would still recalibrate the microbial communities. The body would simply continue to be what 100,000 years of female biology has made. It. The science is a gift to those who want it. It is not a requirement. The body works on its own. But for those who do want to know what they are carrying, we have now offers something that previous generations of women did not have. It offers a way to recognize the signals from your own gut as real signals with deep biological meaning rather than as noise to be dismissed. It offers a framework for understanding why your digestion changes across the month, across the years, across the major transitions of female life. It offers a context for the food sensitivities, the immune responses, the hormonal shifts that the medical system has often struggled to explain. It offers, in the end, a way of seeing your body as something that makes sense, that has reasons, that has a history, and that has been doing its work faithfully since long before you arrived in it. The bones at Sungir lie back under the soil now, after the brief moment of their study. The cave at Hyonim has been backfilled and quieted. The brick factory at Sungir was never built where the burials were found. The mounds at Indian Knoll have been preserved against the river that almost took them. The mud brick walls at Çatalhöyük are slowly weathering under the Anatolian wind. Each of these places held for a time a woman or a community of women whose bodies told us what bodies are. The places will outlast all of us, by some measure. The bodies will not. What lasts is the body you are in. What lasts is the gut that is still doing the work this evening, that women's guts have been doing across 4,000 generations. I want to ask you, as we close this, what part of your own digestive life now makes sense to you that did not make sense before? Tell me in the comments. I am not asking for engagement. I am asking because the answers you give will help shape the questions I take up next, and because the women who hear those questions deserve to be heard by science and by each other more fully than they have been heard so far.